She's listed as being from the Mossad. She was quite keen to point out that she's actually from the Technion, so you don't have to be afraid. Anyway, so please, thank you.
So hi, I'm from Odette Beja's lab, and I'll be talking about type 3 rhodopsins. I hope I'll convince you they're type 3. Well, our lab does discovery-oriented science as opposed to hypothesis-driven science. We try to stay as loosely based as we can. So my objective was to discover new light-responsive proteins, completely new ones. And the currently known ones can be divided into two, either chlorophyll-based, such as in photosynthesis, or retinal-based, such as in our eyes and in microbes. So these are the chromophores which are used by these systems. So type 2 rhodopsins you have in your eyes currently right now, looking at me, you have rhodopsins. And rhodopsins of a similar structure also exist in microbes, as you know. So there are 7 transmembrane proteins, which can either pump ions across the membrane, or they can interact with another protein inside a cell. And so they're very exciting because they actually opened up a new field called optogenetics. You can actually introduce these proteins inside neuronal cells, and then it serves as an optical sensor which generates action potential. And this is a beautiful example of how something that was actually 40 years of microbiologic finding from the environment actually ended up at the frontier of neuroscience. And so what we wanted to do is use functional metagenomics to find new light-responsive proteins. So we had to choose the function which we will screen for. So one of the things that happens with rhodopsins is when you express them inside E. coli and you shine light on them, they sometimes change the pH of the medium they're inside. So you can look for a pH change in response to light. Like here in the 2000 science paper by Beja, you can see here that when you turn the light on, the pH drops, and when you turn the light off, the pH stabilizes. So that's one function. Another feature is when you express them inside E. coli, they actually change the color of the E. coli. The E. coli becomes purple or red or orange. So we devised a screen. You can see here 8 very narrow pH electrodes, and we have light coming up from under, and we shine light on random DNA libraries expressed in E. coli. And we search for either a pH drop, which only happens with light, or we look for colorful colonies. So this is the screen. And then we had to choose what metagenome to screen for, which environment. So we actually chose the lowest freshwater lake on Earth. You might know it as the Sea of Galilee. It's where Jesus walked on water. It contains huge amounts of uncultured microorganisms, of course, and it's under the sun. During August, it was very hot and very sunlit. And we just generated random libraries. So how do you know that you have a jackpot in functional metagenomics? How do you know that you actually found something new? You have a phenotype, okay, we have a color. Then you sequence the clone that gave you the color. So either one of the genes is annotated to cause this phenotype. Maybe it's some kind of a pigment. Then you go like, okay, that's not very important. But if none of the annotated genes there give you an explanation, then you know you found something new. Because it's not annotated, it's never been found before by anyone else. So this is what happened to us. This clone here had purple color with retinal, and no sequencing. Sequencing showed no rhodopsins or anything that explains the color. So we had to know which gene exactly gives us this phenotype. So we did TIN5 mutagenesis, and we had one candidate. When we put it inside annotation databases, it didn't give us anything, but topology predictions told us that it's a 7 transmembrane protein, just like other rhodopsins, but the sequence was completely new. Also, another interesting feature is when we put it inside these prediction topology servers, it showed us that it's completely flipped. it's 180 degrees flipped, as opposed to the type 1 and type 2 rhodopsins. Usually rhodopsins have the N-terminus outside the cell. The new rhodopsin had the N-terminus inside the cell. So we named this new rhodopsin Heliorhodopsin. So their structure is similar. The sequence is different. They all use different types of retinal, and they're completely flipped from others. So when we took this gene and we actually blasted it against known databases, we discovered it was not a single occurrence. It was actually a huge family. We had many, many instances of this protein. And it was almost as abundant, as diverse as the type 1 rhodopsins, which are very diverse. Also, when we looked at metagenomes, we saw that it is almost as abundant as type 1 rhodopsins here in gray, and the Heliorhodopsins, the new ones, are in purple. And sometimes in fresh water, they're even more abundant than type 1 rhodopsins. So actually, you might have it inside your databases right now, and you might even have it inside your cultured microorganisms, and I would like you to be aware of it and maybe search for them actively. So the Heliorhodopsin, what do we know about it so far? It's a membranal protein which incorporates all trans retinal. It has an inverted topology as opposed to the known rhodopsins. It is widely distributed among marine and freshwater environments. And the question is, what does it do? Okay, we know it's a light sensor, we know it senses light. What does it do? So one of the clues about it is the extremely slow photocycle. A photocycle is measured from the time the photon hits the rhodopsin to the time it takes it to come back to its original state. So very slow photocycles are usually indicative of the fact that they need to interact with another protein to carry out their function. So there might be another partner and a transducer involved in this. Another interesting feature which might give us a clue is the fact that it's always never expressed in D-derms. So you can divide the microbes into those that have a single membrane or a double membrane. So when we have a double membrane, you never have a Heliorhodopsin. It seems that it needs to be in interaction directly with the outside world. So this also could have a clue about this. So if you have an idea why that happens, I'd love to hear it. And this is another small slide about unpublished data. We found some more actually pigmented things in our libraries. So we had several type 1 rhodopsins. Here we have the Heliorhodopsin. And here we have an interesting clone. It gives us an orange pigment. It seems like it's a biosynthetic pathway of retinal, because some creatures actually produce their own retinal. But when we sequenced it, we see no biosynthetic pathway of retinal. So we might be onto a new pathway. So stay tuned. So I hope I convinced you that a novel type of rhodopsin was discovered here by functional metagenomics, which is a really beautiful way of dealing with the world of in the world of annotated and misannotated databases. When you try to find something new, you need to step back from the bioinformatics, because the bioinformatics always rely on previously annotated things. And if you think that functional metagenomics is something that might be interesting for you and you want to talk to me about it, I would love to hear your ideas. And thank you so much for the organizers and the funding things, our collaborators from Japan and from NIH, and our beautiful, amazing lab, and especially Odette Beja for this. And thank you.
We can take a question.
Hello, mainly from Shenzhen University. Recently, I found that your group also published one paper on the bioarchive, mentioned the Asca acuta also contained this rhodopsin. So can you provide anything, say something about that?
I mean, with a Czech...
Yes, the Asca acuta.
Yeah. Well, actually, he found there something very interesting. It seems that he found from metagenomes something that is a mix between a type 1 and a Heliorhodopsin. It's probably, it may be something, the missing link in the evolution of this transition from the type 1 and type 3. We don't know. It's very new. It was published in bioarchives, I think, last week or something. It's very interesting. We don't know.
Okay. Any other questions? All right, then I'd like to thank all the speakers this morning. We have another afternoon session, and I hope you can all be back.